Could there come a day when an optimal diet could be recommended, not just for the population overall or for people with special conditions such as diabetes, but a diet that would be unique for you? A diet based on your genetics, let's say, on the condition of your microbiome, perhaps, or on your environmental exposures or other factors. This futuristic possibility may be closer than you think, thanks to the work of researchers, including today's guest, Dr. Stephen Zeisel, Director of the Nutrition Research Institute and Director of the Nutrition Obesity Research Center at the University of North Carolina. I'm Kelly Brownell, Director of the World Food Policy Center at Duke University. Welcome to the Leading Voices in Food. Steve, it's a real pleasure to have you with us.
Kelly, it's a pleasure to be with you as well.
I would like to lead off by asking what you mean when you use the term precision nutrition.
Precision nutrition is really the use of modern day science to be more precise about our recommendations for any given individual. In the last decade, we've realized that people vary greatly in their metabolism, and many people have roadblocks at certain steps of metabolism.
How far along is the science on this?
Well, I think that in terms of the roles of genetic misspellings on your metabolism, we're really quite far along. Now, when I say gene misspelling, each of us has a genetic code, and it's constructed of 4 letters. We inherit our genes from our ancient ancestors, and during many thousands of years, we have developed alternative spellings in some of the genes. I'm going to call them misspellings, called SNPs, SNPs. And these alternative spellings sometimes are in genes that are critical for steps in metabolism and cause us to be more or less efficient at these steps.
Steve, can you give us an example, perhaps even hypothetical, of how such information might get harnessed in order to help a certain person? Would a person have a biological deficiency in some way that could be learned from genetic tests that then could be turned into recommendations for specific foods?
Yes, so again, from my own research in this nutrient choline, choline is really important during pregnancy to build a healthy baby with optimal brain function. Women were designed to be able to turn on the production of choline in their liver when estrogen levels, the hormone that rises markedly during pregnancy, when estrogen rises, they turn on their production to make this choline and therefore are less dependent on their diet. And what we've discovered is that women who come from European backgrounds have very commonly a misspelling in the gene that needs to be turned on, and the switch for estrogen isn't working right. And so they, like men, don't turn on this gene. And this is really important because these are the women who have to be really careful about getting a good source of this nutrient during pregnancy because they don't have the backup insurance of being able to make it by themselves.
Are there other things like environmental exposures that might lead to specific nutrition recommendations for particular individuals, or maybe the condition of the microbiome, or what other sort of factors might be relevant other than just pure genetics?
Genetics is the most ready to go to market because we know so much about these tools. But we differ in our metabolism for other reasons than having genetic mistakes. For instance, our microbiome, which are the bacteria and fungi and other microbes that live in our gut, sees our food often before we do. And if you have a microbiome that is destroying some of the nutrients you need, then you have to eat more in your diet. Alternatively, the microbiome can make some of the nutrients that you need. For instance, vitamin K, very important for normal blood clotting. Vitamin K is made by the microbiome, as is folate, and if your microbiome isn't working well to make it, then you must eat more in your diet. Microbiome is another area in which people can differ, and that can make them need more or less of other nutrients. Environment probably also can change your requirements. We know, for instance, that there are environmental agents that simulate exposure to estrogen, and this can change genes that depend on estrogen and thereby change metabolism. And other foods you eat obviously can be manipulating your metabolism. Grapefruit changes the metabolism of many drugs because it turns on the liver's metabolism system. And what is changing drug metabolism is likely also changing metabolism of the normal components of food that these systems were meant to degrade and excrete.
So this work is very sophisticated and extremely interesting and promising. What do you see as commercial applications for this kind of work?
Right now on the market, there are a number of gene tests, for instance, that tell you how fast you metabolize a component of your diet, caffeine. And there are fast metabolizers and slow metabolizers, and it turns out that the fast metabolizers chew up caffeine quickly, and therefore the person has to stand in line at the coffee shop to get a second coffee, while the slow metabolizers, after drinking some coffee, keep their caffeine levels up for a longer time and don't crave more caffeine so rapidly. That's an example of a gene test you can have to find out if you're a fast or slow metabolizer of caffeine. Of course, most of us know that by how much shaking we do as we stand online at the coffee shop.
So let's turn back to the issue you mentioned earlier, your own work on choline. So you mentioned that it was particularly important during pregnancy. Is it important other times of life, let's say for children?
Kelly, when I started out in my science career, we thought that people didn't require choline in the diet. All the textbooks said that. And in fact, we weren't giving it to people when we fed them intravenously in the hospital, and those people got sick. And when I first started out, I thought it was strange that animals required choline in their diet, but people didn't. And so I put people in the hospital, I took choline away, and what I discovered is that most men and most postmenopausal women, when deprived of choline, developed liver problems, fatty liver and liver damage, or developed muscle problems with breakdown of their muscles. And I found that premenopausal women in North Carolina, only about a little less than half of them got sick when deprived of choline. And so choline is really important for maintaining normal liver function and for maintaining your muscle. The premenopausal women that got sick all had gene misspellings that made them unable to turn on choline formation in their liver with estrogen, and so they were reduced to the sad state of men and had to eat choline in their diets at all times or get sick. So that work proved that humans required choline, and it led to a recommendation by the U.S. National Academies of Science that all humans get it, and we set dietary requirements which are now on food labels. People should eat 0.5 gram of choline a day as adults. A little more when you're pregnant because you use a lot of choline to deliver it to the baby. The choline is used to build the baby's membranes that surround their cells and to build and drive normal development of the brain.
What foods provide choline?
Choline is found in fatty foods often that also contain cholesterol. It's in the membranes of the food, so eggs and meats are excellent sources of choline. Plant-based sources are harder to find, and you have to choose those plant sources that have lots of membranes like wheat bran. For people who want to find out what foods that they eat are the best for getting choline, the U.S. Department of Agriculture maintains a database with thousands of foods in it and tells you what the choline content of those foods are. In the 1970s and 80s, when we recommended everybody cut back cholesterol in their diet to prevent heart disease. The bad part is that we thought we were very smart as nutritionists and nutrition scientists. When we did that, we also cut back choline in the diet without knowing about it. We discourage people from eating foods like eggs. And now choline intake is a problem nutrient in the United States, and every year the U.S. government conducts a national health and nutrition survey, and they find only 7% of women who are of pregnancy age are eating the recommended intake of choline, and at least a quarter are eating half the recommended intake. And that's true for men. The only group that seems to be eating an adequate amount of choline are young children who are drinking lots of milk.
You mentioned muscle function. Could you tell us more?
So in adults, when they're deprived of choline, about 10% develop breakdown of their muscles every time they exercise. And we see that by looking at an enzyme that's released from muscle into blood when the muscle breaks down. This breakdown can be quite severe in some people who are fed a low choline diet, and we find that all of these people have a genetic variant, spelling difference, in the gene that transports choline into the muscle cell and in the gene that first uses choline to make the products needed to help the muscle function normally. We can do a genetic test, predict who those people are, and when we feed them adequate amounts of choline, they do not break down their muscle when they exercise. And this has been of interest in the armed forces, where they find that up to 25% of warfighters who are in training have to stop training because muscle breakdown is excessive. And they're wondering whether some of this breakdown is due to these people who have this genetic difference and a higher need for choline.
So, thinking of the general public, are there groups of people, do you think it would make sense from a public health point of view to be tested for choline? And how would people know what their optimal levels are? What would you see happening with that?
I think that in women who are going to get pregnant, who are pregnant, where choline is so important for building a normal baby's brain, in mouse models we can show that the stem cells that are going to form the nerves within the brain don't divide properly if choline isn't available at a critical time during pregnancy, and the mouse baby is born with many fewer nerve cells in their brain and don't function as well. They are worse at running mazes and at doing cognitive testing. In humans, a group at Harvard School of Public Health looked at women's intake during the first and second trimester of pregnancy and found that women in the lowest choline intake versus women in the highest intake, when their children are studied at 7 years of age for how well their brain performs cognitive function, they find that the women in the lowest intake had children who perform worse at 7 years of age than women in the highest intake. In addition, neural tube defects and other birth defects that are fairly common during pregnancy are increased among the children of women who were on the lower choline intake. And in a study in Berkeley, California, they reported that these neural tube defects, defects of formation of the spinal cord and brain, were increased fourfold in the women who were in the lowest group for intake of choline. And this is after correcting for the vitamin folic acid that we know is so important during pregnancy as well. Women during pregnancy, I think, should have a test looking for these gene differences. And if they have them, they should eat a diet more carefully that contains foods that are rich in choline. Because such a test has not been available, I work with a company that's trying to spin out a gene test. We also are working on fatty liver. About a third of the population in the United States, as they gain more weight, develop excess fat in their liver that seems to interfere with how they use insulin and makes them insensitive to insulin, and this is called metabolic syndrome. And again, people, when deprived of choline, develop fatty liver, and we find that this is because they have a problem sending fat outside of the liver to feed the other organs in the body. And so fat backs up in the liver whenever they make more of it because they're slow to mail the fat out of the liver. And this is related to having trouble making the envelope needed to mail the fat from the liver because that envelope is made of a choline and some other constituents. And so people deprived of choline develop this. People who have metabolic roadblocks in the metabolism of choline and related nutrients have this same roadblock and problems in exporting fat. And when they gain weight, they develop fatty liver more rapidly. We're hoping to develop a genetic test that identifies who these people are, and then we can give the nutrients beyond their roadblocks in metabolism and try to return them to normal.
Thank you so much for sharing your knowledge with us today. I appreciate you joining us. And thank you for listening. If you'd like to subscribe to the Leading Voices in Food podcast series, you may do so at Google Play, at Stitcher, at Radio Public, or Apple Podcasts, or by visiting our website at the Duke World Food Policy Center. This is Kelly Brownell.